This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1145, recorded on August 23rd, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi, everybody. It's a glorious day. 99 degrees. That's not 100. And if I look at the long-range forecast, I don't see any hundreds. We just came through a few, and that was enough. So I'm hoping we're past the peak here and headed on down. It's also bright blue. Good day in Austin, Texas.
Here in New Jersey, it's 41% humidity. It's low. 28C or 82F. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 79 Fahrenheit, 26 Celsius. Partly cloudy sky, but just a really nice day here, too.
Partly sky. There's not a whole
Partly
sky.
Partly cloudy, partly blue.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And here in my office, it's no sky. But outside, it is absolutely glorious and has been for days, and is apparently going to stay this way for a few more days. Same as Kathy, 79 Fahrenheit, 26C. Humidity's 42% here, and just lovely.
If you enjoy these conversations about science, we would love to have your support. Microbe TV, production company for all 10 podcasts, is a nonprofit. So your contributions in the U.S. would be federal tax deductible. You can go to microbe.tv/contribute. We don't take ads. We don't have any support of any kind except yours to make this work. It's the way not to have any conflicts of interest. A little bit of news before we get into our papers. An article from Times of Israel. And it is called UN Agencies Wage Uphill Battle for Vaccination After Gaza Baby Found to Have Polio. So we've talked in the last few weeks about how the unsanitary conditions in Gaza have led to circulation of polio virus in wastewater and sewage. And now we have an unvaccinated child contracting polio. And this is expected because when there's always circulating polio virus, and if you don't vaccinate, which is the problem here, right, given the conflict and everything, you will have polio virus. You will have poliomyelitis.
So
10-month-old baby.
Leading up to this, we've had reports of virus, but no cases of paralytic disease.
There have been no polio cases in Gaza, poliomyelitis cases for 25 years, but they have detected, and it's vaccine-derived type 2 poliovirus, but it's still poliovirus. It still paralyzes. It still transmits. And if you don't get vaccinated, it will cause paralytic disease. So,
and what under these circumstances, what do they do for vaccine? Do they do oral polio vaccine, OPV?
well, there's a picture here of a child getting an injection in his or her thigh. So that would be IPV.
Mm-hmm.
And they say this is a pentavalent shot protecting against 5 diseases. They don't say which, but I'm assuming this is the
DTaP
with
IPV. Yeah,
which would be all 3 serotypes of polio.
that's correct.
Okay. Good.
And just a reminder for the listeners that if there's a case of paralytic polio, the rate of that is typically, depending on which strain, 1 in 100 or 1 in 200. So that might mean that there's 100 other children infected that just didn't show the paralytic signs.
Right. Now, according to this article, it says 1.6 million doses of the oral vaccine would follow. The cold chain components like refrigerators, which arrived Wednesday. So it sounds like they're going to give them OPV, which is unfortunate. I don't know. Since it's type 2, I'm assuming they're going in with NOPV2. And we'll just reseed
Gasoline on the fire. Yeah,
I mean,
but Well, okay, not gasoline on the fire, but it's
The long term is dubious. The short term, yes, you immunize kids and they don't get polio, but then you still have more virus circulating. IPV would be perfect, but I can understand, immunizing so many people with needles is
I
hard.
just
well,
want to. I
and
just want
in a
to.
war zone.
Yes.
I want to check in with newer listeners and make sure they're okay with the nomenclature here. OPV is oral polio vaccine. That
Mhm.
is the stuff that's taken orally. It is a live attenuated vaccine. And although it's a very good vaccine, the disadvantage of that is that you do shed virus, and sometimes or frequently virus that is increased in neurovirulence relative to what went in orally. You shed it in your feces, and that is one of the contributing factors to the reservoir of poliovirus that is still out in the wild. Note that the virus that is has cropped up in Gaza is vaccine-derived poliovirus. Okay, so it's cheap, it's quick, it's easy to take. It has many advantages, good immunity, but does have that downside. IPV is inactivated poliovirus that's taken by injection. It is not a live virus. It works well. It has some of its own disadvantages, not to mention not the least of which is that you got to inject it, okay, and it's more expensive. But it does not have this major disadvantage of contributing to the circulating virus. So there's no one great solution to any of this. And one of the things that this news article does to me is it highlights that, at least for the foreseeable future, this is not going away. Okay. Gaza is just this great example. If you have a healthcare crisis, then something that you haven't seen around for 25 years pops up.
Yep. And
Just a-
this IPV is the standard of care. It's what kids get throughout the developed world.
Yep.
Everywhere there's a functioning healthcare system and functioning government, pretty much everybody uses IPV for exactly the reasons Rich just outlined. And OPV is now used. OPV used to be standard lots of places. It is now only used in poor countries and places that can't distribute IPV.
Just to put this in perspective, we have circulating poliovirus here in the U.S., and when vaccination drops, as it did in Rockland County 2 summers ago, we had a case of polio in an unvaccinated individual. So you have to keep immunizing. The U.S. uses IPV, as Alan said, many other countries do. You have to keep it up. You cannot stop, ever.
Yeah, which is one reason it's so good that in the U.S. and many other places, we now have these combined vaccines like Tdap, where your kid is getting vaccinated at the age that they're getting this. You obviously want to still protect them against tetanus and diphtheria and pertussis, and along free for the ride is all 3 serotypes of polio.
Right. Okay, next up, an article in SIDRAP. More global Mpox spread as clade 1b is confirmed in Thailand, the second case outside Africa. Last week, we talked about the first case outside Africa in Sweden. Now we have a European traveler from Africa to Thailand infected by clade 1b. That's the virus that has been spreading in DRC and neighboring countries. And so this is starting to raise the anxiety level globally. But again, and Daniel talked about this extensively yesterday on the clinical update, this requires close contact in order to transmit. So it's not anywhere near SARS-CoV-2, right, in terms of its ability to infect a lot of people. And, people are always saying, should I get vaccinated? Not every American, not every person in the world needs to be vaccinated. You have to decide your risk factor, for this. Probably if you wrestle, you should get vaccinated.
Yes, I would recommend that.
And others as well. Any thoughts, Rich?
Nothing new. To me, among other things, this is something that can be controlled by appropriate information and public health measures.
Right.
It's a disease which, as far as I know, has little, if any, asymptomatic transmission. It's by very close contact. I don't quite understand why it's showing up so frequently in kids, other than talking about parenting, okay, where the parents have it and they have very close contact with their kids, and the kids are probably obviously a susceptible population. But the way the last global surge, was taken care of was by making sure that the people who were in the high risk populations knew what to look for.
Yeah.
I mean, vaccines, the vaccination obviously helps as well. But this has to do with paying attention, having appropriate information distribution, and taking appropriate precautions as much as anything else. And as Vincent said, this is not anything like SARS-CoV-2. The reason I smile a little bit here is that anytime we talk about, it's probably not that big a deal, I think about the first couple of weeks of SARS-CoV-2, and I shrug.
And when Rich says the first couple of weeks, he means like December of 2019.
Yeah, absolutely.
Well, actually, the second week in January, I
Yeah,
was
it's-
just up on the statistics of that because I
Ah,
presented it. No, yeah, we talked about SARS-CoV-2 on every episode in 2020 except the January 5th episode.
That's
Okay.
right. It hadn't hit yet. Yeah.
Yeah, and
That's
the
right.
my first reaction was, this will blow over.
Blow over.
And then I think the next episode, it was like, this is looking a little concerning.
And I was just having this thing this morning. This is kind of an aside, but every now and then it dawns on me again that these are just little pieces of RNA or DNA. Okay. and they just wreak havoc.
Yep.
Just a little piece of RNA. Give me a
Amazing.
break.
It's amazing.
Unbelievable.
Next up, from the FDA, they have approved and authorized updated mRNA vaccines for COVID-19 to be the KP.2 variant. In June, they had said, we would like you to make the JN.1. And before they could even release that, now they say, nope, we want it to be KP.2. And so that's what's approved. But what's interesting, Novavax is still making JN.1, and that's okay with the FDA. So that's interesting. I don't know what the compelling information is that we need to have KP.2, or this means we're going to update it frequently. So anyway, I don't know when this is going to be available, but
They say maybe as early as this weekend.
this weekend.
Mhm.
So if it's your first vaccine, you have to get multiple doses, but if it's not, you just need one dose of
And
this.
this is monovalent, right?
This is monovalent.
They're not doing bivalent or
Nope.
multivalent vaccines anymore.
No, they did that once, and that was not a good idea, right? Because they had the original plus the
Right.
circulating, and then most of the response was to the original. So it's monovalent. So there you go from the FDA. And interesting article in the Lancet.
Very interesting.
Effectiveness of recombinant VSV ZBOV vaccination, that's Ebola virus, during the 2018-2020 Ebola virus disease epidemic in the DRC. This is a very interesting test design. It is a retrospective test negative study. And so it's very cool the way they do this. So this is all over this epidemic, right? So they're using data, and they did vaccinate during the epidemic. So they've collected data, and the people who are participating in this evaluation, well, their records are participating in this evaluation. They had a PCR result and were eligible for vaccination and had symptom onset with Ebola. And so each individual is confirmed by RT-PCR to be Ebola virus disease positive. So that's a case, and each one was matched to one individual negative for Ebola virus disease. That would be the control, because you can't do a case control because the thing is over, right? and they match the individuals by sex, age, health zone, month of symptom onset. And then they calculate the effectiveness from the odds ratio of being vaccinated greater than or equal to 10 days before symptom onset versus being unvaccinated. So that's the case negative. And here are the results. So they had 1,273 of 26,000 individuals were positive for Ebola virus disease, and 25,000 were negative controls. 40.
So that's about 5% of the eligible were positive, and about 95% were negative.
Right. And so from those numbers, they calculate that the effectiveness of this vaccine against Ebola virus disease is 84%. It's darn good.
Mhm.
Yeah.
Darn good. What I think of when I read about this trial and these results is that this was really an emergency program swung into action. This was a SWAT action, okay? Because this broke out, and it was a striking epidemic. If I'm not mistaken, this particular recombinant virus, and this is a real recombinant virus vectored vaccine. Okay, there are
Right.
not many of those out there, and this was, I think, the only VSV platform recombinant vectored vaccine that I can think of. Okay, so vesicular stomatitis virus, which is not completely it's a mononegavirales, right? Okay, so it's not completely off the wall relative to being related to Ebola, but with its own surface glycoprotein having the Ebola virus glycoprotein spliced into it. Okay, I think that was at least through preclinical trials, if not phase 1 testing, so it was around. Okay, but they cooked that up and said, this is our best shot here. And went into a population where the disease was already raging. Okay, they couldn't set up, the standard. Let's get a bunch of volunteers and that kind of stuff. And so I forget the details of how this trial was structured, but it was very interesting the way it was done on a population that already had disease running through it. And and it seemed to be effective just anecdotally as you looked at the impact on the disease, but now we have real data. Very cool.
This was first tested during the 2015 West African Ebola outbreak. It was a phase 3 cluster randomized ring vaccination. We talked about this because the name of the trial was Ebola, ça suffit, it's enough. And vaccine efficacy was 100%.
Yeah.
We talked about it here, and then I did a TWiV in Rotterdam with a lady from WHO who had designed the trial, and it's really remarkable. So that's the vaccine that is now used, was used in this house. So 84%.
Sorry.
No, it's okay. That's it.
Another good example of not pandemic, but preparedness. Okay.
Mm-hmm.
Okay, finally, last one, CIDRAP. Georgia reports another case of measles, and there's an outbreak in Oregon, 30 cases. So the Georgia patient was exposed during international travel, and then the Oregon, the largest outbreak since 1991. All patients were unvaccinated. 2 were hospitalized. 12 are younger than 10. 11 are 11 to 19. Really, parents, you don't vaccinate your kids against measles.
I had I dug into this just a little bit, and the issue here, the problem here, is that Oregon has one of the highest rates of vaccine exemptions. And
Yes.
some of the loosest laws covering vaccine exemptions around. And one of the articles that I read contrasted Oregon with California, where some time ago California was having a similar type of problem, And they said, okay, enough of this nonsense. The only way you can now be exempted in California from vaccination is if you have an absolutely legitimate medical excuse. None of this funny business that basically amounts to, I don't want to vaccinate my kid.
Yeah, I think California, like many other sensible states, you need a doctor, an actual board-certified physician to write a note explaining why you're not being vaccinated.
So this outbreak illustrates why school mandates are needed. If any of you have read Project 2025, one part of it says the CDC will have no role in making recommendations for vaccination. It will be up to parents and medical providers. This is what's going to happen if you let that occur, because parents and medical providers cannot look at the trials and decide whether to vaccinate or not. We talked about this on Beyond the Noise with Paul Offit the other day. He said, the clinical trial data for the 2 mRNA vaccines, 900 pages. Parents and medical providers will not read it. They'll just go to Google and they'll get some anti-vaxxers who say things and they say, oh, I've done my research and I'm not vaccinating.
Parents in particular. Yeah.
Parents in particular.
Well, it would be a very interesting nationwide study on how this works out.
Well, there's an easy way to have to leave the CDC. By the way, the CDC, the FDA, and the NIH are all targets of Project 2025. There's an easy way to make sure it doesn't happen, folks.
Yes.
You have something called a vote. All right, time for some science. I don't like to mix science and politics, but it's already mixed.
Well, yeah.
There was, I think, is there a letter coming up in the queue? I saw
Probably.
something recently about us doing politics and basically our attitude towards this is if politics stays out of science, then we'll stay out of politics.
Yes.
But there are times when the politics impacts on the science that we're talking about. Okay. Yeah. And at that point, it is it's more than appropriate. It's a responsibility to respond.
All right, some papers we have for a snippet, something very different from the usual. There is a virus involved, but it largely has to do with development, and it's in this. My light just went out. I guess it's not plugged in. All right, we'll have to be Rembrandt style. Okay, no problem. Cell Stem Cell is the journal, and the title is A Primate-Specific Endogenous Retroviral Envelope Protein Sequesters SFRP2 to Regulate Human Cardiomyocyte Development.
We have 6 co-first authors.
Yep.
Ran Zhang, Menghua Wu, Dan Zhang, Jieying Zhu, Qi Zhang, Hui Zhong, and the senior author is Dongwei Li. They are from Guangzhou Medical University in Guangzhou, the Chinese Academy of Sciences in Hong Kong, University of Macau, Guangzhou Institute of Biomedicine and Health, and Southern University of Science and Technology in Shenzhen, Westlake University in Hangzhou, and the University of the Chinese Academy of Sciences in Beijing. Oh, and also Macau University of Science and Technology in Macau.
So we're going to have to come back to the meaning of the title in a second, but I want to be a little bit pedantic here because this comes up in this paper and also the second paper. And it just bugs me when they say things like, I'm just going to shorten it here, a protein sequesters another protein to regulate human development. Okay, it doesn't do it for a purpose of. And using the word to connotes that to me almost every time I see it. And so if they would say the protein sequesters a protein, comma, regulating human development, then that would solve the problem. But it's attributing, in a way, some kind of function or choice or something that isn't really there. It's a complication of the English language, and so I'll get off my pedantic pedestal.
That's quite all right. Yeah.
Anybody want to make a quick summary?
Well, there are 3 main proteins involved. One of them is this retroviral protein. Then there's this one that Vincent likes the name of, this secreted frizzled protein.
Mm-hmm.
And then the third one is the I guess
Wnt.
it's it's beta-catenin, right?
Yeah.
Yeah,
Yeah.
Wnt beta
Wnt.
catenin. Yeah. Yeah. So this is an endogenous retrovirus. So it's a remnant of an old retroviral infections of primates, and the protein is still being made, and it's involved in the development of cardiomyocytes, cells within the heart, as you will see. So we know that these endogenous retroviruses, so when retroviruses infect cells, their RNA genome is converted to DNA. It integrates into the cell, and if the cell is a germline cell, then it stays with the organism, is passed on to offspring. And humans have somewhere between 5 and 8% of their DNA as integrated copies of various endogenous retroviruses. And over the years, they've been studied, and people are finding out that they have, in some cases, functions.
I like the
The-
line where they say this is endowed organisms with a genomic landscape rich in viral relics.
Rich in viral
And
relics.
we're talking, when we talk about long ago, in this particular case, and in many cases, we're talking about 50 or 60 million years ago.
Yeah.
So
Yes.
that's even pre-primates, right? That's
Yeah.
so early
Oh,
in
yes.
vertebrate evolution.
Oh, yeah.
So
I like-
typically these integrate and they become inactive, and they're not producing any proteins, and they're just sitting there in the genome, possibly serving some function, but we're not sure what. A few of them do produce proteins, and some of those proteins have been implicated in very early stages of development, like formation of the placenta, which we've talked about on TWiV in the past.
Yeah, syncytin is the famous one, derived from a retroviral envelope protein. So retroviral viruses encode 3 main genes: GAG, structural proteins; PAL, the polymerase and other proteins; and the envelope, which is the spike glycoprotein. And so the envelope has been repurposed to make syncytin, which is important in development of the placenta. This paper talks about a endogenous retrovirus, ERV, called H48-1. And it has been suspected to have other functions before. It actually may regulate fusion during placental development, may also restrict infection by other retroviruses. But protein coatings have not been implicated in embryonic development before. So they want to study that here. And this HERV H48, it first invaded the primate genome 40 to 60 million years ago. And so they compared the sequence of this ERV, this 48-1 among hominids and old world monkeys, and they all have a conserved 160 amino acid gene. And so they said, okay, maybe there's something going on here. So they looked at transcriptomic data from a variety of embryonic and adult somatic tissues. And they find it at very low levels in most places, its expression, except in human pluripotent stem cells, right, which are the cells that give rise to pretty much all the other cells. That's why they're pluripotent. So they say, 48-1 may have a role, maybe in human embryonic development. And then they say, human post-implantation embryonic development is not easily accessible for analysis. Yeah, it's not.
No.
You can't do experiments on embryos, but you can do single-cell transcriptomics, and a lot of that's been done, and you can access the data. And so they looked at it and said, where is 48-1 made? And they find that it's not only expressed in the trophectoderm. I'm going to test your cell layer knowledge here, and placental tissues, but also in the embryonic endoderm and mesoderm, both in vitro and in vivo. So this is just by mining RNA-seq data. So
And
they said,
these, so we're going to be talking about ectoderm, endoderm, and mesoderm. And as the roots of the words imply, the ectoderm forms your skin. It also forms your neurons. The endoderm forms things like the insides of your intestines and so forth. The mesoderm is everything in between, which is producing a lot of stuff.
so the 3 layers, they all give rise to the entire embryo, basically,
Yeah.
right? And those come from pluripotent stem cells.
So let me even go back to my embryology a little more and test myself out. So we're talking about shortly post gastrulation,
Yes.
right? So you start
Mhm.
off, an embryo starts off as a sphere, a ball
Mhm.
of cells. And
Right.
then that goes through gastrulation, which means that at some point on that sphere, there's an invagination, okay, that
Kathy is doing a
folds.
motion with her fingers for those of you on
Okay,
the
good.
video.
That folds part of this inward and then closes up. So now we have uh, two layers of cells and a layer in between, basically, what was the internal part of the sphere. And those basically translate, don't they, into ectoderm from the
Mm,
outside,
that's right.
mesoderm, and endoderm. And you get differentiation beyond that. And I looked up because I was very curious. They were very straightforward in saying, this is not easily accessible material when you're talking about human material. And I looked up the at least one, I think it was the paper that they referenced for single cell transcriptome analysis, and it was from an elective termination of pregnancy that went through all the informed consent and everything else, and the investigators had a gastrula, a human gastrula, to work with, and went through and did a lot of single cell transcriptome analysis on that.
Mm,
And they've starting off with mined those data.
got it. So remember now, we're talking about a single gene from this endogenous retroviral, 160 amino acid gene. So they say, let's learn about what this gene is doing in development. So they knock it out in human pluripotent stem cells. And then they look at the transcriptome to see if anything is affected. And it's not, barely perturbed. So then they say, okay, let's take these and differentiate them into mesoderm and endoderm lineages, which you can do separately. And they find that markers for those lineage cells are reduced by having no 48-1 in there. So they say, oh, this means that somehow we're disrupting the specification of these lineages. So 48-1 must be involved in this differentiation. Okay. So then they differentiate the pluripotent stem cells to cardiomyocytes. They decide to explore the role of this in cardiomyocytes. I don't know where they got to
I'm
cardiomyocytes.
guessing, so I got to this too, and it was, you go to endoderm, and then next thing you know, it's cardiomyocytes. That was oddly specific. And
Yeah.
I'm guessing they either had insight from literature that they haven't cited here, or um, that they looked at other types of cells and didn't see anything interesting, and the cardiomyocytes looked messed up, and
So
so
they
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 pursued
do
them.
find that the protein is made throughout cardiomyocyte differentiation. So that
Yeah.
maybe was a hint. So they can differentiate their human pluripotent stem cells to form cardiomyocytes in vitro, right? And if you knock out this 48-1 gene, so if you differentiate HPSCs to cardiomyocytes, within a couple of weeks, you can see beating cells in the culture.
And by the
This-
way, just to bring people up to speed on this who may not be familiar with the pluripotent stem cells, these are cells that are taken just like you can get them from yourself from a cheek swab or something, any somatic cell. And by treating them, as was discovered some time ago, you can treat them with specific factors that will cause them to regress back
Mhm.
to what they were in early stages of embryonic development, and that turns them into these stem cells. And they are pluripotent because they can take many different subsequent developmental paths. And in the past several years, people have worked out how to induce them to form different things. And this is actually why we have a journal called Cell Stem Cell, because this is really cool, and there are a lot of people working on it.
So those would be induced pluripotent stem cells,
Right.
iPSCs.
Yes.
I don't know if they use that here because in some countries you're allowed to take, embryos and make
Right.
HPSCs from them. So But
But
yes,
the iPSC procedure is so well documented at this point. I think that's probably what most people are doing.
yeah.
And so in terms of the issues that we've talked about of accessibility and ethics, okay, this is clean. They're
IPSCs,
using,
yeah.
there was that one thing that was done quite a while ago that we already talked about, which was transcriptomics on a human embryo, but that's now all on a computer. And the rest of this is cell culture. Now, the cell culture models are good. They're not perfect. It's good to have the human samples to compare them to, but it does significantly get around the
Yes, the ethical
ethical
issues.
and
Yep.
accessibility problems.
What's amazing is, Alan said, I think it's 3 plasmids
Yeah.
you put into any cell encoding 3 different regulatory proteins, and it turns them into a pluripotent stem cell. It's
It
a
is
Nobel Prize
shockingly simple now that
winning
it's known.
discovery. It's
Yes.
amazing. Maybe it's now 2. I don't even know, but it's not a lot.
Let me interject one thing here that I'm not sure about, but you guys can check me on. Throughout this, I was thinking of papers that, if I'm not mistaken, there is a burst of sort of a global burst of ERV transcription
Mhm.
early in development, right?
Yes.
And there's
yes.
always been the implication from that maybe these ERVs have something to do with early development and differentiation. And now it's going to turn out we have an example of that.
Yeah.
Hold on.
Right. Vincent's doing some tech stuff here.
Okay. So you can differentiate these iPSCs into cardiomyocytes, and they will beat in the culture, which is really cool.
And there's videos with,
Yes.
with this article, and it's open access, right?
Yeah.
And
Both the papers
then
we're talking about today are open access.
if you knock out the 48-1 ERV gene, you don't get beating cardiomyocytes. It's a pretty cool phenotype, right? It's very
Yeah.
clear. So somehow this protein is playing a role in the cells going from HPSCs to cardiomyocytes, right?
Now,
And
if this was a fly genetics paper, they would have renamed ERV-48 Tin Man or something.
Yeah. Yes. It would have given it a good name. Now, getting from HPSCs to cardiomyocytes, there are multiple steps involved. There's the pluripotent stage, the mesoderm stage, and then the final generation of cardiomyocytes. So they tested at the role of 48-1 at each step, and it's involved in all of them. Okay. So that's pretty cool. Now, you're going to go off after I plugged you in, huh? What the hell is the story here? You see? It is plugged in. Okay. All the stages of transition. Next, we're going to introduce some other kinds of genes here. So what's going on with the knockout? What's happening to these cells? So they do a time course in the knockout cells, and they say by day 7, the knockout cells are not committed to going to cardiomyocytes, but instead they go to a different kind of cell population that expresses ectoderm marker genes, which is not the right one for cardiomyocytes. So somehow the presence of this, in the absence of the gene, you're going in the wrong direction. So somehow 48-1 keeps the cells going towards the cardiomyocyte. If you take it away, they diverge to what they call an ectoderm-like fate. All right, so then they say, let's make sure this protein is made in these cells. And so they do mass spec. It's funny that we're a quarter of the way into the paper, and they just so they do mass spec, and they they do find the protein in human pluripotent stem cells, and in fact, they find part of the protein, the N-terminus of the protein, has a number of hydrophobic residues, which they say maybe it's a transmembrane domain or a signal peptide, right? A signal peptide, of course, is at the N-terminus of proteins. It helps the proteins get through membranes So they're thinking maybe this protein is membrane associated. So they do a series of experiments by labeling the protein 48-1. They show that, in fact, it's membrane associated. And if they take off the N-terminus with the hydrophobic sequence, the protein no longer goes towards membranes, and it loses its ability to participate in cardiomyocyte differentiation. So this N-terminal hydrophobic sequence is really important for making cardiomyocytes, right? So it has a signal peptide that directs the protein to membranes, and that's important somehow in making cardiomyocytes. So they're thinking, well, maybe it's binding another protein, right? Maybe it's getting secreted and binding another protein, and that's how it's working, because signal peptides often direct proteins to the secretory pathway. So they do protein-protein interaction to look for anything that will bind to the 48-1. And they get 27 candidates, and they find one which seems to be co-localizing with 48-1 in cells, and it's called SFRP2. SFRP2, which is secreted frizzled related protein number 2.
Speaking of fly geneticists.
Love frizzled. And this is a known protein that regulates stem cell development, basically. It's known to regulate stem cell development, okay? And it's actually been linked in mice to cardiomyocyte generation. And so they do a series of experiments looking at the co-localization of these two, and they conclude that 48-1 binds to SFRP2 and sequesters it in a membrane compartment and limits its ability to be secreted. So SFRP2 would be normally secreted. 48-1 keeps it in a membrane compartment, and that allows cardiomyocytes to develop. If you without 48-1, SFRP2 is secreted, and it inhibits the transition to cardiomyocytes.
And if that happens, then the differentiation goes down a different path to
That's right.
ectoderm, which in their graphical abstract they show as the SOX2.
Right.
Right.
And not only does it 48-1 sequester the SFRP2 in a membranous compartment, but it seems to help get it degraded.
Tags it for degradation, yeah.
Mm-hmm.
It targets it for degradation by targeting it for what we call polyubiquitination, attachment of small ubiquitin moieties on which target it for degradation. And so that's how it is regulating it. Now, it's interesting. There's no 48-1 in mice.
Yeah.
This is a human. It's a primate.
As I was reading this paper, I was like, how is IRF48-1 is a primate
right
virus, and everything else in our line develops hearts and certainly mesoderm, and how do they do it?
Right. So mice have SFRP2, and it's very similar to human. So they do an experiment and show that, in fact, IRF48-1 will bind mouse SFRP2. So they say could be there's something similar in mice that has a similar role, but it just hasn't been found yet, right?
MERVs.
MERVs, yes.
I want to interject here for a minute. Once again, I'm thinking about people who haven't necessarily been with us that long, and about these phenotypes and their funny names. Okay. One of the earliest model organisms for geneticists was the fruit fly Drosophila, talking about early 20th century, among other things, because you could make variants of the fruit flies that had really obvious phenotypes. You could see something wrong and you could breed these and they would breed true. And the Drosophila geneticists had a sense of humor, okay? But also there were pretty straightforward descriptions. And so we're going to see two of these in this paper. One is frizzled and the other is wingless, okay? And I just looked up frizzled. The frizzled locus of Drosophila coordinates the cytoskeletons of epidermal cells, producing an array of cuticular hairs and bristles. In frizzled mutants, the orientation of individual hairs with respect to both their neighbors and to the organisms is altered. Okay? In the typical wild type wing, all hairs point towards the distal tip. But immediately you can see that there's a connection here, that these are developmental issues. Okay? And if you tinker with these proteins, you get developmental phenotypes.
The very first fly, mutant fly, was made by Thomas Hunt Morgan in the early 1900s at Columbia University. He just looked at thousands and thousands of flies and found one with white eyes. And it was a little sickly, so he had to nurse it and breed it. And eventually they figured out they could use mutagenesis to make mutations, right? Like kind of UV and other things like that. But that was the first one. It was brute force. Very cool. That's all in the book by Jonathan Weiner. What's the name of it? He talks all about the development of genetics over the years. I can't
I'll
remember.
look it up.
It's not the Beak and the Finch. It's another one. Okay. Here's a really cool experiment. All right. So you knock out 48-1 in human PSCs and you don't get cardiomyocytes, right? You take those cells and you knock out SFRP2. What do you think happens? You get cardiomyocytes.
You get cardiomyocytes.
Because it's the SFRP that is no longer being retained by 48-1. And now when you take away SFRP2, you get normal development. It's a beautiful experiment, right? And it confirms the sequence of events. I like that very much.
Is this time, love, and memory?
Time, love, and memory. Exactly. And I remember this Thomas Hunt Morgan story. His wife had a baby. She was in the hospital. He went to visit her, and she said, "How's the white-eyed fly doing?" He didn't want it to die, right? Because he only had one. Okay, so 48-1 sequesters SFRP1 in membranous compartments. It's degraded. You need this signal peptide domain for that. And so they transfer the signal peptide from 48-1 to SFRP2, and that rescues differentiation in knockout cells, because now the SFRP2 normally is going to the membranes because it's got a signal peptide that you've put on it from 48-1 just to make sure it does all the right things. And now you get cardiomyocyte differentiation. So this is like fruit fly experiments in development.
In human
right?
cells.
Very
Yes.
cool. It's very cool. Let's see. I think there's one more experiment here. So they say, okay, so all this suggests that 48-1 is needed for the mesoderm and also the endoderm. So they differentiate their PSCs into mesoderm and endoderm, and they look at wild type and knockout cells, and they show that basically both lineages are affected by 48-1. So it's a lovely example of how a viral protein has taken over a role in the cell in development, right? So it was there as a consequence of infection. It was in the germline, so it remained, and over time it acquired another function, which may be random. It has something to do. It has a signal peptide, so it's in the membrane. The ability to bind SFRP2. I don't know if that arose or if the ancestor had that ability. We don't have the 40 million year old virus, because it would be nice to have that and know if it interacts with SFRP2.
Well, I mean, the little bit that they did with mice hints at the alternative approach, which is to take extant species and survey them to see how they deal with this problem, and see if there are other ERVs there, and if so, is there some reason that the primate line might have switched to this one, or is this one better at it, or What?
Now the axis here that's involved, we haven't mentioned it, but the developmental axis that 48-1 is involved with is also involved in differentiation of osteoblasts, chondrocytes, adipocytes, so lots of other different cell types. So it could be that this 48-1 is involved in those. And so they end up by saying that this endogenous retroviral gene has implications for regenerative medicine. Maybe we can manipulate stem cells as part of if you have an issue with a tissue, you can make stem cells and manipulate 48-1 and make cells that can replace yours. So that would be cool, right?
Yeah.
And so this is one of many, many thousands of endogenous retroviruses in us, and maybe the others also have roles. It's just a matter of looking for them.
They're
Right?
just hiding there in the genome all this time.
Yeah. So that's not a virus paper, but it shows you how viruses have been essential for development.
I
mean,
think it's a virus paper.
A virus is the star of this paper.
Virus is the star.
Yeah.
If you didn't have this virus, we'd have no hearts.
That's right.
We'd be heartless. Without viruses, we'd be heartless. Okay, now a bona fide virology paper. This is a Nature article. Shifts, and I learned a new word here. Shifts in receptors during submergence of an encephalitic arbovirus. And this comes from a group mainly at the University of Texas Medical Branch Galveston, but also Harvard Medical School, and Galveston, Rich and I visited a couple of years ago. And in fact, one of the authors here, Scott Weaver, we did a podcast with him and a few others. First author is Wan Yu Lee, and the last author is Jonathan Abraham. Any takers for a summary?
Also collaborator from the Broad Institute and Brigham and Women's.
Any summary?
Well, I won't do the summary, but I do want to comment on the word submergence. I bet 99% of the people who see this in the title will have to look it up. And basically, it's the opposite of emergence.
Yeah.
So,
yep.
So things emerge means they come up, they come into being, they become evident, and the opposite of that is submergence. They're here and then they go away.
I can have a crack at a summary, at least prime the pump. So this is about Western equine encephalitis virus, which is a toga virus, an RNA virus that is also it's insect vectored. Its ordinary life cycle is to be shuttled back and forth between mosquitoes and birds. Okay, so it spends part of its life in mosquitoes that then bite the birds. The birds are infected and make more virus. The mosquitoes suck that up, infects the mosquitoes, etc. But the mosquitoes also bite other creatures, including, in this particular case, notably horses and people. And the virus can cause a disease in those creatures, though they are dead-end hosts, which means that they don't cook up enough virus to be transmitted back to the mosquitoes. That infection is done. This particular virus was first uncovered in the early 20th century, around 1930, with an outbreak in the southwestern United States, California, that killed like 700 people. Was it 700 people or 700 horses?
6,000 encephalitis cases in horses, and they don't have the human casualties here. But it was a bunch of people died.
Enough
too.
to get people's attention. Okay.
Yeah.
And then over time, in the ensuing 100 years, it seems to have, in the human and horse population, kind of gone away. And so the question becomes, but the virus is still out there, or circulating in mosquitoes and the birds. And so the question becomes, what's happened? has there been some sort of evolution of the virus that has reduced its virulence. And they investigate that here. And part of that investigation involves identifying the receptors for the virus that gets it into cells. And what they discover is that there has been a change in the receptor specificity of the virus, so that whereas the older versions of the virus used to use a receptor that was available to them in both humans and horses, the newer versions of the virus don't use the receptor as it's presented in humans and horses. Okay, so its virulence apparently, it's over. Virulence is a funny word in this case to me. Okay, but in terms of the epidemiology, the virulence appears to have decreased because the virus just isn't infecting those creatures anymore. And as long as I'm at it, I want to read this cool thing that I found in Wikipedia about the discovery of this. Western equine encephalitis was discovered in 1930 when a number of horses in the San Joaquin Valley of California died of a mysterious encephalitis. Carl Friedrich Meyer investigated but was not able to isolate the pathogen from necropsies of horses that had been dead for some time and needed samples from an animal in the earlier stages of the disease. When the team heard that a horse had appeared to have encephalitis, its owner threatened to shoot the scientists. However, Meyer was able to convince the farmer's wife that the horse was dying anyway and secretly signal him when the farmer was asleep in exchange for $20. This was during the Great Depression, and that was a substantial amount of money. Meyer and his colleagues hid in the bushes until the signal euthanized the horse and stole its head.
God.
They successfully isolated WEEV from the brain tissue.
Stole its head. Did they put the head in bed with the
It's not
farmer?
right. I was just thinking of the Godfather.
It does conjure up the Godfather, doesn't it?
Well, now the farmer's going to discover
He's
this.
probably going to notice. Yes.
I also
Going to ask his wife, "Do you know what happened?" And she'll say, "I have no idea."
I don't know.
yeah, either that or she gave him the $20 and said, here
This
you
is
go.
what happened.
This
Oh,
will
wow,
help
that's
you
funny.
out.
Let's buy a couple of new horses.
I also, in looking up to make sure for myself that the humans and horses were dead end hosts, I poked around and I found some verbiage verifying that from an organization called the World Organization for Animal Health that I have to say is the acronym is WHOA.
Whoa.
I love it.
It's cool.
Okay. There you go.
All right. Thank you.
Sure.
All right. So these are alphaviruses, right? And we have weave, Western equine, and we have Eastern equine and Venezuelan equine. And they are all involving mosquitoes, avian reservoirs, and humans and horses are dead-end hosts. We get the infection from mosquitoes. And you can get mild or asymptomatic infections, or you can get encephalitis, which, of course, is very serious and can have long-term sequelae. We have no vaccines. We have no antivirals against any of these. And as was already said, we first isolated in 1930. There were many thousands of cases in horses. There were subsequently many thousands of human cases. But since the middle of the 20th century, the weave outbreaks in humans and horses have declined in frequency and scale to the point where also seroprevalence in places like California has decreased, and surveillance in birds and mosquitoes showed a decline in circulation, the submergence, right?
Mm-hmm.
So, and they say this is also accompanied by a decrease in the mammalian virulence of contemporary strains. So those viruses isolated in the 30s and 40s are more virulent in mouse models than virus, say, isolated in 2005. And so that's in the U.S. Similar things have happened in South America, although there was in 2023 a weave outbreak in Argentina and Uruguay with many cases and fatalities. So, this decline is not absolute. Can be exceptions, and that we get at what might be happening there in this paper. So okay. So we have
Even I would like to point out, even though there have been these outbreaks like in Argentina and Uruguay, um, and with human cases and human fatalities, the human fatality rate seems a lot lower than it used to be.
yeah.
So the kind of the top line here was, could be 50% lethal, and now it's like 10% in the Argentina outbreak. So
Yeah.
the lethality of the virus in humans does seem to have declined.
So there are two groups, two
I still wouldn't
lineages.
want a 10% lethal virus, just to be clear. That's
Yes. No, you don't want that. So there are two groups or two lineages, group A and B. And B is divided into B1, B2, and 3. So group A has the ancestral strains, which are highly pathogenic, the original isolates. B1, 2, and 3 have moderately pathogenic strains, and B3 at least one that is non-pathogenic in animal models. And the later viruses have apparently displaced the earlier ones, so that now today in North America, B3 is the only one that we can detect circulating in North America. So they look at receptors basically in this paper to try and see if that answers what's going on in the epidemiology. So as we just talked about recently, they take cells that can be infected with TWiV. They do a CRISPR-Cas9 knockout strategy, genome-wide strategy, and find cells that are resistant to infection and then ask what gene has been disrupted in them. And they identify a gene called PCDH10, not frizzled, unfortunately, but PCDH10, which if you knock that out in 293 T cells, they become resistant to infection. And then if you put the gene back, they can be infected again. Antibodies against PCDH10 will block infections. And if you have a cell line that doesn't have the receptor naturally or PCDH10, if you put it in, the virus can now infect those cells. So it looks good that this is involved in infection. What is PCDH10? It is a transmembrane domain with 6 extracellular, what are called cadherin repeats, number 1 through 6, and they do some modification of the gene, and they find the cytoplasmic domain is not essential. The transmembrane segments are not, but it's the first cadherin-like repeat. If you just make that on cells, it will allow the virus to enter cells. So apparently you don't need the rest.
I want to interject a couple of comments here. First of all, the cadherin is a one of the, now I'm blanking on the generic word, but it is a protein that's is expressed on the surface of the cells and helps cells stick to each other and to the extracellular matrix. And this one is particularly important in forming synapses and neuronal development. And the cytoplasmic domain, there's communication between the outside of the cell and the inside of the cell via the cytoplasmic domain. That's, I don't think, entirely relevant for this, except that virus does cause an encephalitis, right? So it's using a receptor that is in neural tissue and in the brain. The other comment I wanted to make was that this is just a general comment. This is one of the few papers from recent times that I could basically read by going through and looking at the figures. And I really appreciate that. It helps that it's closer to my, usual reading and easier for, I'm more familiar with it. But it's really well written, and the figures are really easy to follow.
So, and these domains, the one that's numbered 1 is farthest from the surface of the cell. So it's
this protein, this first domain, if you make the protein the first domain only, it will bind virus particles. They do some live cell microscopy that shows that the virus can bind. They have fluorescently labeled virus particles, and they show that with PCDH10 on the cell, the virus will bind and gets taken up into cells. So this looks like a bona fide receptor for at least weave. Okay, so when you do phylogenetic analysis of the part of the virus, the structural protein, to see what's binding the receptor, the spike protein is composed of two subunits, E1, E2, and it looks like the E2 has the sequence that binds cellular receptors. And so if you look at different isolates over time, there are changes in here. And they want to know if these changes have happened that could affect receptor binding. So they make viruses with E2 domains from different strains representing groups A, B1, B2, and B3. So 14 different E2 sequences. And then they have cells producing PCDH10, and they say, can these viruses all bind to PCDH10? They also use cells that make very low density lipoprotein receptor, right? So that's been shown to be a receptor for some viruses. And in this case, it is a receptor for Eastern equine encephalitis virus and ApoE-R2, which is also a receptor for that virus. So they include those in their analysis. And all the group A strains up to 1941 can bind PCDH10, VLDR, and ApoE-R2, all three receptors, to infect K562 cells. So the older strains can bind all three of these receptors. And that's part of the big story here, as you will see. They looked at B1 and B2 strains and the South American strain that we talked about. They recognize PCDH10, but not VLDLR or ApoE2. And group B3 from 1985 and 2001 recognized PCDH10. Two group B3 strains did not, though, from 2005. And so these are binding assays. So they also do infections in cells. They have bona fide replication competent virus. Sorry, those are virus-like particles, I believe. So they do it with bona fide virus, and they can show that the ancestral viruses, the older viruses, replicate faster into higher levels when you have PCDH10, VLDLR, and ApoE2 present than in control cells. They also do, you can make soluble versions of these receptors and ask, will they block virus entry into cells? So if you have cells making PCDH10 and you use a soluble VLDLR, it will block all the A strains from entering via PCDH10. So having low-density lipoprotein receptor bind to the virus particle also will block them from getting. So it must be some common area of the spike that is involved in receptor binding, right? But in the same assays, if you use a group B2 strain, which will only bind PCDH10, the soluble low-density receptor protein will not affect that binding. So these different viruses over time have different receptor specificities, as you can see. So then they say, what if we disrupt either PCDH10 or VLDR on entry for the group A strains? So they do that. And the California, Macmillan, and CPA viruses, these are old viruses. They are blocked in PCDH10 knockout, but not in VLDR knockout cells. So it looks like they depend on PCDH10, at least in certain cell types, and have similar efficiencies of infection. Okay, now they ask, what about virulence? All these proteins, PCDH10, VLDLR, and ApoE2 are on central nervous system cells. So they get cortical neurons from mice who have been knocked out for the PCDH10 gene. And they infect with group A viruses. And they find that the viruses can infect neurons, but not the knockout neurons. They can infect wild-type neurons, but not the knockout neurons. So PCDH10 is absolutely essential for infection, even though the other proteins are present in these neurons. Sole determinant of neuronal. These are strains that only recognize PCDH10. Let's see what else here. Don't want to confuse people.
So right, these were they also found that the older, let me see if I've got this right, the older virus spike proteins could infect the PCDH10 knockout neurons, right?
Yes.
Not as well as if it had PCDH10, but they could still infect.
And you know what the problem is? They, instead of using group A and B, and they give them the real names like Macmillan.
Yes.
I don't remember.
Yeah,
so Macmillan is an old one,
yes.
1941. So the earlier ones, Fleming, California and Fleming, these, and 71V. What is 71V?
That's a group B2 from 1971.
So B2 can infect neurons, but it needs PCDH10, because if you knock out PCDH10, it won't infect them. But Macmillan, as you just suggested, Alan, infection is reduced but not abolished in PCDH10 neurons. So it's probably using those one of the other two receptors to get in. Those older strains can do both. And so that the point here is that it happens in neurons as well as in other cells in culture. Right? So PCHD10 and LDLR family receptors support neurotropism of the Macmillan strain, and that 71B, a B2 strain that doesn't bind LDLR, requires PCDH10 on cortical neurons. Okay, now what about challenge in mice? So the results so far say that group A strains recognize PCDH10 and VLDLR through overlapping surfaces on the spike. Remember, we did that experiment before. So they take the Macmillan strain and they challenge mice with it. It's lethal in mice, but if they treat mice first with soluble PCDH10, they prevent lethality. So Macmillan can engage multiple receptors, but blocking its ability with PCDH1, soluble PCDH1 blocks its ability to get into all of them.
This is reminding me of the criteria that there are if you want to establish that something is a receptor. And one of them is if you can take that molecule and block infection, then it helps solidify your conclusion.
And of course, here they're doing cells in culture and animals, which is good, right? So then they look at other animals, other non-human mammals, birds, reptiles, and amphibians, where this protein, PCDH10, is highly conserved. They check horses, house sparrows, which are a reservoir of these viruses and also amplification hosts. So next time a sparrow is flying around you, it could have weave in it. Garter snakes, which are supposedly overwintering hosts, right, when there's no mosquito around. I guess mosquitoes bite snakes, right?
Sure.
Why not?
Yeah.
Snake can't do anything.
No.
Snake can't.
That's right. It can't swat it away.
These receptors from PCDH10 from all these species allow virus to infect which virus? Group A, Macmillan, and group B271V. So all those other species receptors works for those viruses. They also found that two group A weaves can infect cells with the VLDL orthologs of horses, starlings, and mosquitoes, as well as ApoER2 orthologs of mice, horses, and starlings. Group B weave strain 71 didn't recognize any VLDR or any ApoER2 of any of these species. So that's a more contemporary strain, right? So it's lost the ability to bind to those receptors. And then they do a series of experiments with a group B3WiV called Imperial 181. It was isolated in 2005 from mosquitoes in California. It doesn't cause any mortality in mice or hamsters. It does not recognize PCDH10, VLDLR, or ApoE2. So, but this virus will grow in house sparrows. So they cloned the house sparrow, PCDH10, and put it in cells. And guess what? The imperial virus will grow in cells with the house sparrow. I was going to say horse sparrow, with the house sparrow receptor. I think that's pretty cool. So, in other words, if a virus has lost the ability to bind a human receptor, it may still be out there circulating in another species, right? That's what the cool part of that is. So, all right. So, what does all this mean? PCDH10 is a receptor for weave, but apparently a recent isolate has lost the ability to recognize that, but that doesn't mean it's gone. It could be in house sparrows. And they say it could reemerge if it changed again to be able to recognize human PCDH10.
It could also be in other birds
or other birds
not
as
just
well.
the house sparrows.
I'm picking on the house sparrows.
Well, it's what they happen to look at.
They look
They're
at,
easy
yeah.
to find. Yeah.
Yeah. And all of us see them outside all the time, right? House sparrows. The ancestors of weaves that caused a lot of outbreaks, they bind PCDH10, VLDLR, and ApoE2. And more recent isolates have lost the ability to bind VLDR and ApoE2, which could be related to their submergence, right?
So this is an interesting evolution.
Very interesting.
Yes.
Because, the receptors haven't changed. I'm a little surprised, maybe we'll see it in the future, that there isn't a bunch of alpha fold or whatever associated with this to try and pick apart what has changed about the virus attachment protein that can now discriminate between the sparrow receptor and the human or horse receptor.
It doesn't take much, right? It could be 1 or 2 amino acids.
No.
Yeah.
And
So-
I actually found the evolutionary aspect of this fascinating because
really interesting.
domestic horses are an introduced species in North America. There were equids in North America before the last ice age, and then they went extinct. They persisted in Europe, and then they came back with colonists. And so these animals didn't exist in that range. And then we had presumably a spillover event in California that got it into the horses from the birds.
Right.
From the horses to the humans, maybe that was the order of events. So that was all a sideshow for this virus. Its normal cycle was always in the birds. And now we see that it has subsequently evolved to be less virulent and perhaps less infectious to horses and humans, simultaneous with horses becoming a lot less prevalent in that area because they're no longer, people use tractors and cars.
Right.
So that back in the 1930s, there was still a lot of horse use for transportation and motive power. But that's now horses are very expensive luxuries, and there are a lot fewer of them.
Yep.
And they're also vaccinated.
Right.
There's a weave vaccine for horses. So that has gone away, and the virus has evolved. They comment in this paper, it's not clear if this evolution of the virus was just something that happened because there isn't any selective pressure in favor of neurovirulence, or if it's something where this change in receptor preference gives it some advantage in the sparrows and the garter snakes that allows it to persist better in the environment. If it's the latter, then that's good news because that would suggest that it'll persist. If it's the former, that this is just a lucky coincidence, then we really ought to keep an eye out. I mean, in any case, we really ought to keep an eye out for the reemergence of this more broad-based receptor
Right.
preference that could produce another neurovirulent spillover.
It's important to recognize that humans and horses are irrelevant to the natural history of these viruses because they exist in enzootic hosts, birds, for example, and they that's where they maintain themselves. And we're accidental hosts,
Yeah.
and we don't
Dead end
transmit
hosts.
Dead end
ends.
hosts, even.
So
Yeah.
it doesn't matter if we get infected or not. If we don't get infected, so some change occurred, probably to improve fitness in the enzootic host, and it destroyed it destroyed their ability to infect us. It's no problem. They're still going to exist, right?
So playing
good for
reviewer
us.
number 3 here, I want to see not only the AlphaFold and the modeling at least, but also the Biacore analysis with the strength of the interaction between the modern virus attachment protein and the various receptors to address the question that Alan brought up, whether in fact there have been changes that make it better, more fit to infect the birds. And as a consequence, we've lost the ability to infect the humans and the horses.
So they make a statement here, which I think is really good. Whether strains that cannot bind human PCDH10 have become dominant in North America awaits further sampling. We don't know. Because fitness of weave for transmission by mosquito vectors among avian hosts has not significantly decreased, strains that can recognize human PCDH10 may be poised for reemergence. So those viruses are still circulating unchecked in mosquitoes and birds, the enzootic hosts or epizootic hosts. And so as long as, if it's 1 or 2 amino acids, it could just randomly happen, right? And then you have reemergence in people. So you need to have sampling, right? Environmental sampling to see what's going on. And if you find in Western Massachusetts that all of a sudden the viruses can bind PCDH10, then problem.
Yes. That would be WMEVE, not weave, Western Massachusetts equine encephalitis. So it also occurred to me, we may have triggered this in some way. The house sparrows that they, Sturnus vulgaris, I should search that up. So is this the European sparrow? Yeah, European starling. Wait, Sturnus vulgaris is a starling, not a sparrow. What's this? Starlings. Yeah, okay, they mentioned starlings. Yeah. So this starling is an introduced species in the U.S., and it is now a reservoir host, apparently a significant reservoir host for Western equine encephalitis virus in California, which may have applied evolutionary pressure to the virus to adapt better to this other introduced species in which it can actually propagate.
Yeah.
That's a theory, but it's one that you
It's
might be able to
interesting.
test if you can find more wild hosts and see if it's, better adapted to the starling than it is to some native species like the garter snakes.
And as far
I
as
think
we
the garter
know,
snakes are native.
the birds are okay with infection.
The birds don't seem to care. This
They
is
don't
not
seem to
West
mind.
Nile.
Yeah.
Yeah.
So their last sentence is really good. Outbreak preparedness could be bolstered by future studies to identify the key sequence polymorphisms in the weave spike, E2 and E1, that explain shifted receptor compatibilities. Because you want to know what to look for in Western Massachusetts, right? Or elsewhere.
Or elsewhere, or New Jersey, or Michigan, or
Because every
Texas.
change is not going to allow binding to PCDH10. You want to know the ones that will, okay? So that's the coolness of this paper. Okay.
Very good.
I like it.
Neat stuff.
Let me look here briefly. I think we have, we should do some picks because these letters are long, right?
These are fairly long letters, yeah.
So, yeah, let's go right to the picks. Kathy, what do you have for us?
I'm not there yet. Oh, okay. So I had someone send me email And I couldn't figure out anything about this person because they had a Gmail address. So I typed their name into Google to see what I could find. And I found two people with the same name, a father and the son, who were quite wealthy, well-to-do. One was the founder and owner of a large company, and the other was a owner of a major league sports team. But I don't know if either one of them is who emailed me. And so the hint for people who write to me is, if you want to get a response, give some context of who you are, or I'm not going to answer, because I won't get as much in the future. But I have gotten email, because I'm with ASV, and I get email because I'm with TWiV. And if it's just something out of the blue and it's something that I don't really want to delve into, take the time, then so, I'm not going to do it unless I know a little more about you. Okay.
Mm-hmm.
Anyway, so what this led to is that I had just heard something on the podcast that I listened to called Away with Words. And I put in a clip from there where they talk about what Google gangers are. And Google gangers is a play on the word doppelganger, from the German words that literally means double goer. And it's a Google ganger is two people with the same name. And so in that Away with Words clip, they talk about some Google gangers, and these two names that I found who wrote to me would be Google gangers, although they're a father and a son. But there could be many other Google gangers relating to this person who sent me email. So
I
know.
love this term. I used to have a page on my website, a disambiguation page, like they do on Wikipedia, for all the other Alan Doves on the internet, because I would get notes from people, and I still get notes occasionally from some, insurance company or something in California saying, here's your new policy. I'm like, no, that's the Alan Dove who's a lawyer in California. I'm the science writer in Massachusetts. You got the wrong guy.
yeah.
And I'm not the former go-kart racer, and I'm also not the jewelry store manager in England. And by the way, I'm also not Al Dove, who is a marine biologist. Yeah.
I have found at least two Vincent Racaniellos.
I was going to
in the
ask.
US.
That's interesting.
Yeah, I remember that you
Yeah.
have talked about that. Yeah.
In fact, one of them lives in Harrison, New York, and he wrote to me because he's a lawyer and he was doing some work for the Columbia Legal Office. And they said, "Do you know Professor Racaniello?" And they said, "Oh." So he wrote me a letter, which I still have. I kept it in a physical letter to this day, because he told me the history of his family. And one of his ancestors was also a Vincent Racaniello. And he said they used to call him Bozeep, which I thought was the funniest nickname. Hey, Bozeep! Can you imagine? But there's a Rich Condit, right? At least
There's
one.
a few. There's the biggest problem is a Richard S. Condit, who is an ecologist. In fact, he's a forest ecologist. He specializes in the tropics, who also, though as a graduate student and after me, went to UC Santa Cruz. Okay. So people trying to figure out what's going on, cross-reference that and confuse us. He actually did his PhD with a guy that I did a project with when I was an undergraduate. So I used to get his papers to review all the time. And so and he used to get some of mine. More recently, I ran into a Rick Condit because he sent me an email that he got asking for a review of a tropical biology paper. And I said, no. That's yet another Rich Condit.
That's a different, yeah.
But this guy is a professor of music and a jazz saxophonist at Lamar University in Beaumont, Texas. Okay, so we had quite a nice correspondence. There are a few others out there as well. How about you, Kathy?
So the one that's been the most annoying for me is that there was a Catherine Spindler who attended the University of Michigan. And once you get a unique name, which is the first, it can be up to 8 letters, and that unique name is what defines your email address. Once you have that unique name, it's with you for life, and nobody else can ever have it. And she chose Spindler. And so the email program now that most people on campus use does kind of an auto suggest or auto fill or whatever. And so there are people who have sent me email, and we find out days, week, months later that, didn't you get that email? And I will say no. And that's because they sent it to Spindler, because it came up as Catherine Spindler. And that person does not respond to that email address, because I have written to her and said, could you put, a message, like an out of office message, only one that just says, I am not the Catherine Spindler who's a virology professor. You want KR Spin, but there's no reply. So
this is a Michigan email address.
Yes.
I think you got to get the university to just nuke the address.
No, it can't be done. And I actually wrote to them once to say, can we find out more about this person? Or do we, and there was virtually no way, but whoever I wrote to said, don't worry about it. I get email from so-and-so all the time. And it was somebody with the same name at the university. So, yeah.
I just Googled Catherine Spindler, and I found someone from South Africa. Have you found her?
No.
She's an artist on artsy.net.
Nice. I'll have to check that out.
But much younger, born 1982.
Yeah. Cool.
That is very cool. I thought my name would be the hardest, but there's probably more than two, actually.
Yeah.
And my grandfather, my paternal grandfather, was Vincent Racaniello in Italy, so there are plenty of them. Rich, what do you have for us?
I don't think I have picked this before. If I have, my apologies. I've gotten so that, you used to keep a list of these things, and I'm not saying that you should, of picks, because you got other stuff to do. And I used to make sure that I wasn't picking redundantly. Now I just Google TWiV and my pick and see if it's there. And usually that works out. At any rate, this is a thing called, I don't know how I ran into this, but I found it at least amusing. RP Flip. And the FLIP stands for Floating Instrument Platform. And this is
But it also describes the operation of it.
Yes.
Mm-hmm. Mm-hmm.
So this is an ocean-going vessel. Says owned and it's actually out of service now, but it was in service for like 40 years, even longer, 40 or 50 years. It was an open ocean research platform owned by the Navy and operated by Scripps Institution of Oceanography. 108 meters, 355 feet long, designed to partially flood and pitch backwards 90 degrees, resulting in only the front 17 meters of the platform pointing out of the water, with bulkheads becoming decks. When flipped, most of the buoyancy of the platform was okay. Its purpose was, to do oceanographic measurements of one sort or another, in particular acoustic research, okay, because the front end of it went way down under the water, and it was insensitive to a lot of the bobbing up and down that would interfere with similar measurements from other vessels. Designed to study wave height, acoustic signals, water temperature and density, and for the collection of meteorological data. So I have a Wikipedia site for this, and you can read about it. Actually, I should mention it did not operate under its own power. It had to be towed because if you're going to do acoustics, it's not a good idea to have a bunch of engines going on. So this wiki site includes a bunch of pictures of the thing, including a video of the flip operation. Okay, it flooding those compartments in the stern and flipping up 90 degrees. So
I wonder if you're on this thing and it's flip, what do you have to do? Right?
You have to scurry around.
Be in the right part of it.
To get yourself in the right part. And there's a little thing in the Wikipedia thing about, arrangements they had to do with the toilets and other pieces of apparatus on the boat. Okay,
It's so
so
So-
pretty
odd.
amusing.
So I have heard about this before, UCSD being my PhD alma mater and, Scripps being part of that. But it does say that the last cruise was 2017, and in August 2023, it departed for a scrapyard in Mexico, apart from an instrument boom, which will be installed on a pier at Scripps.
Yeah.
This
I
thing is
remember
really
hearing
cool.
about I remember hearing about this too because it's so cool.
Yeah.
What it's doing. My
And
gosh.
I think somebody proposed this as a general design for ocean platforms that would be used for other
Uh-huh.
research and that could be used, for a variety of things, but it never really took off.
But it would move around, I presume, right?
It would
stay-
drift with deep currents,
Yeah.
which you could compensate for in various ways, or I suppose you could anchor it. But it wouldn't be affected by surface wind because most of the ship is underwater when it's done its Titanic on purpose type move.
It says here in the entry, the platform was frequently mistaken for a capsized
Yes.
ocean transport ship.
Yes, people would report it.
I can imagine. Oh my gosh. 700 gross tons. That's very cool. I would never have thought of this again, Rich. You brought it back. Thank you.
Yeah, wow. Good fun.
Yeah.
Alan, what do you have for us?
I have a pick it's a recent news article that I thought was very interesting. So we've seen in science in recent years the growth of larger and larger teams, which has some benefits, but it also has some drawbacks. It concentrates money in the hands of fewer PIs, and I think we've talked about this various times on TWiV. Somebody finally did a study on what impact it has on your career if you're part of a small group versus a large group. Is it better to be, one of the 50 people in a big lab or one of the 6 people in a small lab? It's not the way the study was done. I think it's pretty convincing. You could argue that it needs to be analyzed a little bit more, but they looked at career outcomes for people who are on these various size teams across many fields of science and ranging from physics to biology. And they found that, in fact, being on a large team is probably not so good for your career. And being in these large groups is a prognostic indicator of leaving science, leaving the lab, versus being on a smaller team. You're more likely to potentially get a tenure track position later on. This is particularly acute for women and people of color who are on these large teams. Just an interesting data point that I think many of our listeners who are training in various lab settings might want to consider.
I've always suspected this, right?
I have long suspected this, and I just thought, well, how would you look at that? And they took a kind of a clever approach, and I've linked to an article that you can read on Science about how the study was designed and the analysis done.
So it's particularly unfortunate because NIH is encouraging big groups and
Yes.
discouraging smaller groups, and has for some time now. And I don't think it's fair to the investigator's career at all.
And if
No.
you look at the papers that we do, I mean, they have the authorship is huge.
Yeah.
Often,
when
quite
I
often,
was
yeah.
when I was growing up, 4 authors was a lot. And it was more
Yes.
like 1 or 2.
I had two author papers as a postdoc,
Mhm.
and I'm sure Kathy did, and Rich did,
Yep.
and Alan
And
had
in graduate
couple of
school,
papers
it
with
was
me.
you and me, Vincent.
Yep.
It was-
Matter of fact,
I
I
loved
had a 2-author paper as a graduate student that was me as a graduate student and a postdoc, Ed Niles. And both of the PIs left themselves off the paper.
nice.
I think that's a way to. You can't hide in a big group, right? Like Alan used to come into my office once a week, and you
Yep.
either have something or not. And,
Yeah.
I think that's a good way to be trained. I mean, people will say, "Oh, Vincent, that's because you were trained that way." But this paper is really supporting that it's not a good idea.
Yeah.
Actually, this is sort of well, a little peripheral, but I had motivation recently to look up Fred Sanger on PubMed. Okay. Now, here's a guy who won 2 scientific Nobel Prizes, one for figuring out how to sequence proteins and the other for figuring out how to sequence DNA. He had a grand total, once I've weeded out all the Google gangers, okay, he had a grand total, from what I can tell on PubMed, of 85 publications, a quarter of which were single author publications.
Yeah. Yeah, and I do certainly understand that as science has progressed, there is a need for more collaborators across institutions, and I'm fully supportive of that trend, and I think it would be interesting to do another study like this, another analysis like this, comparing whether these were strictly affecting authors in big groups or authors in cross-collaborative groups as well that happened to have a lot of authors on the publication because you had 9 different institutions participating in your, clinical trial. But just this level of it is pretty eye-opening.
The photo on this article is unbelievable.
It's from CERN, yeah. So the big physics stuff, and the photo in the lead is this enormous room full of people.
My God.
Yeah.
They say physics is one of the biggest, had most authors on a journal article.
Yeah.
Well, folks, you have a choice when you're looking for a group.
Mm-hmm.
Always about a choice. All right. So my my article is from Wired magazine by so many ads. Come on. Oh, wait, did I make a reader out of this? No, I didn't make a reader.
Rhett Alain is the author here.
Yeah, Rhett Alain. So you're underwater. Why do you see that circle of light above you? It's called Snell's window. Have you ever gone in a pool and you look up, there's a circle of light, and you can often see things on the side reflect. Well, they have a picture of it right here, right? Sat at the bottom of a pool and pondered your watery ceiling. Most of the surface is a sheet of blue. You can't see through it. But right above you, there's a round window of transparency. This fisheye view shows you not just the sky, but stuff around the side of the pool. It's caused by the optical properties of water. It's called Snell's window. And this is an article that explains it. And I love it that this is a thing that you can see, and they explain it in pretty good depths, right?
Well, it's got math in it.
It's got
math. It's trigonometry basically, but it's interesting math.
I need to go do the experiment. I spent a lot of time underwater, and I've never really, if I've noticed this, I certainly haven't thought about it.
Well, now the next time you go in, you're going to see it.
I'll have a look.
In fact, listeners, they have an experiment here using a laser in a tank of water that illustrates. Anyway, I really liked it because it's a good job. So Rhett Alain is an associate professor of physics at Southeastern Louisiana University. I think the physicists have to be better at explaining things because their stuff is inherently hard, right?
Reminds me immediately of the Einstein quote that if you can't explain what you're doing to a layman, then you don't understand it.
You don't understand it yourself.
Yep.
Yeah. All right. That's TWiV 1145. You can find the show notes at microbe.tv/TWiV. You can send questions and comments and pics to TWiV at microbe.tv. And if you enjoy these programs, we would really like to have your financial support. Microbe.tv/contribute. There are a number of ways you can do that. It doesn't have to be a huge amount. Be a cup of coffee a month. It's tax deductible. If you go to buy a cup of coffee, it's not tax deductible, right? But if you give it to us, it is. Kathy Spindler is at the university. Wow, Kathy Spindler actually is a professor emerita at the University of Michigan in Ann Arbor. I had to get used to doing that from memory, Kathy. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of University of Florida, Gainesville, currently in Austin, Texas. I put you at a different place, Rich. I'm sorry.
Okay.
Thank you.
Sure enough. Always a good time.
And Alan Dove is easy. AlanDove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm also easy, microbe.tv. I'm no emeritus yet. Maybe I won't be. I don't know. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.